Vanakkam friends, Trinity Cultural Academy Coimbatore and New India Publishing Agency New Delhi welcome all of you for this presentation, and the presentation is on male sterility and hybrid development. This comes under the plant breeding and genetics, and this is the sixth series of plant breeding. Now we will go the topic one by one. It is being prepared by me. I am Dr. Vanangamudi from Tamil Nadu Agricultural University. I am a retired dean. And first of all, we will see what do you mean by sterility. So this is a non-functional pollen grain in the female parent. Okay, and this is being coined by Cowell 1988. In other words, it is inability to produce viable pollen due to failure of formation of functional stamens, microspores and gametes. And the actual reason for this male sterility is mutation technique. And here I have given two flower diagram. The left one is the male fertile flower that will give the viable pollens. And if you look at the right side figure, this is the sterile parent. The pollen is sterile, and here the left diagram, the pollens are very viable and the anther lobe is big and other things. Here in the right diagram, this is almost closed. There is no anthesis at all. And if you look at the mutations, the manifestations of male sterility, there are different mechanisms are involved. The first one is absence or malformation of male organs, and it is a failure to develop normal microsporogenesis tissue, and that is nothing but the anther development. And the third one is abnormal microsporogenesis that will give either deformed pollen or inviable pollen. And mostly there is an abnormal development and maturation of pollen grains, and the another manifestation is non-dehiscent anthers, but the pollens are viable, but there won't be any anthesis. And the last one is the barriers other than incompatibility preventing pollen from reaching the ovule. Ovule is nothing but the stigma. The stigma has to reach the, pollen has to reach the stigma, and again it moves as a pollen tube to the ovule. If you look at the history of male sterility, the first report was given by Jones and Eschweller in onion crop. In 1944, Jones has first found S type of cytoplasm from USDA stock, and the scientist Roger, he has found T type of cytoplasm that will give the male sterility. And Collier 1973 observed anther abortion within the species as well as species of maize hybrids. And genic male sterility has been reported in cabbage by Rundfeldt in 1960 as well as in cauliflower, and this was found out by Nifoof 1961. Male sterility system have been also developed through genetic engineering by Williams and co-workers, and particular protoplast fusion by Pelletier and co-workers in 1995. Male sterility were artificially induced through the mutagenesis. There are some mutagen chemicals that will also give male sterility, induce male sterility artificially. And the first time report in maize was by Kullreuter 1763, Beadle 1932 and Roger 1953. They found out cytoplasmic genetic male sterility in maize. Rhodes in 1933, he has found out cytoplasmic male sterility in maize. Jones and Davis 1944, CMS in onion. And we observed different pollination controlling mechanisms like the manual emasculation, and there is a use of male sterility as well as use of self-incompatibility alleles and use of male gametosides. There are different chemicals that will induce male sterility artificially. Use of genetically engineered pollen killer genetic system. These are all the different mechanisms that will control the pollination or pollen production. And if you talk about the benefits of male sterility, and this will help in reducing the cost of hybrid cereal production because there is no manual labor involvement in this type of male sterility. If you take the hand emasculation and the pollination technique in cotton, it involves a lot of labor and more expensive than compared to the male sterile line. And there is a possibility of production of large scale of F1 hybrid seeds, and as I told, it avoids the enormous manual work on emasculation and pollination, especially in cotton. And this type of male sterility will speed up the hybridization program, and there is a commercial exploitation of hybrid vigor due to this male sterility in various crops like rice, maize, sorghum and bajra and red gram as well. And we can create male sterility by different mechanisms like spontaneous mutation, that's a natural mutation, and we will go for interspecific hybridization, and again mutation induction and genetic engineering through the biotechnological tools, and chemically induced male sterility, that is the CHA in short. And we can detect the male sterility by seeing the performance of progenies on a crossing with few normal genotypes, and in which the detection system consists of system 1. Here all progenies in all the rows may be sterile, that is otherwise called as cytoplasmic male sterility. And the system 2, some rows may consist of all fertile, but in some other rows there is a occurrence of the fertile as well as sterile plants in 1:1 ratio, and that is the mechanism is the genetic male sterility. And the system 3, some rows are fertile and some rows are sterile, and some other rows are sterile and fertile in 1:1 ratio, that is cytoplasmic genic male sterility. These are all the three systems available in our breeding program, and this can be utilized for exploitation of hybrid vigor and development of hybrids. And now coming to the classification of male sterility, Cowell 1988, he has classified the male sterility in three major groups. The first one is phenotypic male sterility, that is a morphological male sterility, and the structural and staminal male sterility. Here the male flowers or stamens are malformed and non-functional and completely absent. And the second one under phenotypic male sterility is pollen male sterility. Here the male sterile line that will produce only sterile pollen, and it is isogenic line of the male fertile. And this what do you mean by isogenic line means? Both the male sterile parent and the male fertile parent are looks like same morphologically, physiologically, growth wise, except the pollen sterility, that is called isogenic line. And the second classification is genotypic male sterility, and there are about 3 to 4 subdivisions in this. First one is genetic male sterility, in short we will call as GMS. And there is a sensitive, some of the plants are very sensitive to environment, so that is environmental genetic male sterility. And under environmental sterility there are two types. One is thermosensitive genetic male sterility, in short TGMS. It's completely governed by the temperature. And the photoperiod sensitive genetic male sterility PGMS, it's completely governed by the photoperiod. Those things we will be discussing in the subsequent slides. And there is a environmental non-sensitive type of genetic male sterility is also available. And the second major type in the genotypic male sterility is cytoplasmic male sterility. And this sterility is mostly determined by the cytoplasm, the mitochondrial or chloroplast genes. And this results in mutation in mitochondrial genome, mtDNA, that is a mitochondrial dysfunction. And the progenies would always be made sterile, since the cytoplasm comes primarily from female gamete only. And the nuclear genotype of male sterile line is almost identical to that of the recurrent pollinator strain. And male sterile line, that is the male fertile line, that is the maintainer line or B line, is used to maintain the male sterile line, that is the A line. And this type of mechanism is available in 3-line hybrid seed production techniques that we will be seeing in the next series. And the CMS is not influenced by the environmental factors, either a temperature or the photoperiod. So this type of cytoplasmic male sterility is highly stable when compared to the genetic male sterility, GMS. And this CMS is being completely and fully exploited in the production of hybrids. And this picture explains the different stages in which the hybrids are produced. That is the A line. A line is the female parent, which is sterile. And the B line, it is the maintainer line, and this is fertile cytoplasm. Both are isogenic lines. They are similar to each other except fertility of the pollen. In A line there is no fertile pollen. In B line that is having the fertile pollen that is used to maintain that A is crossed with B to maintain the male sterility that will be subsequently used for hybrid seed development or hybrid development. And if you cross A and the B lines you can get only the male sterile line, and that has to be crossed with the restorer line. Or means restorer line, that is nothing but the fertile line. It restores the fertility of A line, and if you cross A and R line you can get the F1 hybrid. And this is the mechanism of hybrid development using cytoplasmic male sterility. In the subsequent slide, in this slide, I will be briefly explain how the sterility mechanism are being transferred to the strain A by back crossing method that is highly useful. And if you take this diagram, and this strain A is sterile line, okay, and the B line is male fertile line. If you cross you can get only the male sterile line, that is the F1. And by doing this we can restore about 50% of the nuclear genes of F1 would be from strain B. So in F1, and there is a transfer of 50% of the nuclear genes to the sterile line. And this F1 has to be back crossed again with the strain B, that is BC's back cross 1. And by back crossing you can able to transfer 75% of the nuclear genes from strain B, and you have to do repeated back crosses, maybe from BC1 to BC7, and BC1 you can transfer 75%, and after that you have to do 7 back crosses until about 99% of the nuclear gene would be transferred to the line, the sterile line. So the male sterile version of the sterile B. These two are isogenic lines. Okay. Then the genetic male sterility, the next mechanisms the involved in production of hybrids. It is also called as nuclear male sterility, and here the male sterility is governed by single recessive gene. Please kindly make note of this. And here sterility is governed by recessive gene, small ms, but dominant gene governing the male fertility. Dominant gene means capital MS, that is the male fertility mean, and small ms is the sterile parent. And the origin may be the spontaneous mutation or artificial mutation. By artificial mutation you give gamma rays or EMS, that's a chemical, ethyl methane sulfonate. These chemicals are common in creating the genetic male sterility. MS alleles, that is a recessive alleles, this will have a major influence on staminal initiation, stamen and anther sac development. Primary mother cell production is also retarded, and there won't be any meiosis. And if there is no meiosis, pollen formation is affected, and maturation and decisions. The anther may not say descent in the sterile parent. And this is the genetic male sterility. And the different mutagens are used for creating genetic male sterility. I have given few examples. The colchicine that will be exploited in jowar, ethidium bromide that is being exploited in the groundnut, maize and wheat, acetone that is in barley. And there are different types of genetic male sterility, and the first one is environmental sensitives, that is the GMS. This MS gene expression occurs within the specified range of temperature and also the photo period regimes. And this type of mechanism is being used for the production of hybrids in rice, tomato and wheat. Mostly rice is being exploited for this GMS. I mean environmental genetic male sterility. And there are two types, TGMS, temperature genetic male sterility. Here at high temperature, the same plant, it is failed to produce the pollen by way of failure of pairing of two chromosomes at metaphase. So this abnormality leads to abnormal meiosis, abnormal or sterile pollens. So if there is high temperature, there is a sterile mechanism occurs. And the anthers were shriveled and non-dehiscent in this type of male sterility, which is being influenced by high temperature. While at low temperature, the same line produce the fertile pollen. If you grow the rice in a particular temperature, and the temperature and other things I will be dealing in the next video, and the same in the same field, if it is exposed to the higher temperature, that will give sterile pollen, and at low temperature the same plant will give the fertility. And the second one is PGMS, the photo period sensitive genetic male sterility. It is governed by two recessive genes, and under long day condition there is a sterility mechanism. Under short day conditions there is a fertile mechanism. The best example, classical example is rice. The sterile plants, the sterile pollen will be produced under long day, maybe 13 hours 45 minutes of long day, and at the time temperature may be 23 to 29 degree Celsius. But fertility, the fertile pollen will be produced under short day conditions. By using the nuclear male sterility GMS, and the hybrid development is taken place, or the hybrid seed production is done, and this figure will show the how the GMS is being involved in the production of hybrids. This is mostly in cotton as well as in red gram. And now the cotton, the emasculation and the pollination technique is followed, and this type of GMS is being neglected, but it is available in red gram. And here the male sterile one, that is the parent one, male sterile, and the male fertile is the parent 2. This is female parent and this is the male parent. When you cross these two parents, you can get the F1. Please make note of this one. About 50% of the population, that's offspring, is male fertile, and 50% is male sterile. So you have to remove all the sterile parent plants and only to retain the fertile. You have to remove all the fertile plants and leaving only sterile plants in the field. Again, it is crossed with the male fertile line. You can get F1 hybrid. F1 hybrid. And the cytoplasmic genetic male sterility, this is the third type of mechanism available under male sterility, and it is also known as nucleoplasmic male sterility. And here R line, that is the restored gene line, is generally dominant, and that can be transferred from related strains or species. And this technique is mostly used for hybrid development in rice, maize, sorghum, bajra, and sunflower. And this diagram will show how the hybrid seed or hybrids are developed by using CGMS, the same diagram which I have explained earlier. A line, that is a female parent. This is the sterile line, and this is the B line. It is the maintainer line. This is used for as a male parent, and if you cross A line and B line, you can get the male sterile line. Here make note that when compared to GMS, here it will give only sterile plants, whereas in GMS 50% is fertile and 50% is sterile, and 50% fertility plants has to be removed, and remaining sterile parent has to be crossed with the male fertile parent for production of hybrids. That is in the case of GMS. In the case of CGMS, and the F1 is completely male sterile, and it has to be crossed with R line, restorer line, to produce the hybrid F1. This is always fertile. And male sterility mechanism is being used for the production of hybrids in crops like rice. Here I have given the complete list of hybrids, rice hybrids, year of release, duration, yield, developed by which institute or which university, and these hybrids are recommended for which state or different states. I have listed out, I have depicted in this table. Please kindly go through this. And about 30 to 40 hybrids are from state agriculture universities as well as the ICAR research stations located cut across the country. And I have not included the private hybrids in this table. I have only included the public sector hybrids in this table. Similarly, the sorghum hybrids is also being listed out in this table, and you can read all these things. It is available in the PDF, and about 20 hybrids so far released from sorghum, and also sunflower hybrids. Many hybrids are developed by using CGMS. I have given tabulated all these hybrids, available hybrids, commercially being utilized and being released by the public sector companies like the state agriculture universities and ICAR institutes. I have given here. And there another section is the chemically induced male sterility, that is CHA. Chemical that will induce artificial non-genetic male sterility in plants, that is used as female parent, also called as CHA, is also called as gametocides. male sterilants, selective male sterilants, pollen suppressant, polynoside and anthracite. And this was first reported by Moore and Naylor in 1950 by inducing male sterility in maize by using the malic hydrazide MH. And there is a properties of an ideal CHA must be highly male and female selective, should be easily applicable and economic in use. Time of application should be flexible, must not be mutagenetic effect, and must not be carried over in F1 seeds, must consistently produce more than 95% of the male sterility, must cause minimum reduction in seed set, should not affect outcrossing, should not be hazardous to the environment. Some important the CHAs I have given in this table. Zinc methyl arsenate and sodium methyl arsenate is being commercially used for rice. Ethipon or ethyl in barley, oats and bajra. Mendoc in cotton and sugar beet. Gibberellic acid in maize, barley, wheat, rice and sunflower. Malic hydrazide in maize, wheat and cotton and onion. And finally, we have two slides. First of all, we can see the significance of male sterility in plant breeding. So it avoids emasculation in female parent. GMS is being exploited in castor in USA as well as red gram in India. CMS and CGMS are in hybrid seed production of rice, maize, sorghum, bajra, sunflower, sugar beet and as well as some ornamental plants. It saves a lot of time, money and labor. There is a limitations in male sterile line utilization. There is a existence and maintenance of A, B and R line is laborious and very difficult. If exotic lines are not suitable to our condition, the native adaptive lines have to be converted into MS line. That will take lot of labor, lot of money, lot of time. And adequate cross pollination should be there between A and R lines. These are isogenic lines for good seed set. And synchronization of flowering should be there between A and R. Mostly in rice, sorghum and the sunflower, there is a non-synchronization problem is always there because A line is different from R line. So that should not be there by using this male sterility. And fertility restoration should be complete, otherwise F1 seed will be sterile. Isolation is needed for maintaining the parental lines as well as producing the hybrid. Thank you very much for listening my presentation. You can kindly go through all these things, watch this video and get benefited, and we will meet in the next video presentation. Thank you.